Here we are again with the latest news from the website prostatewarriors.com. This deep dive will once again focus mainly on preclinical studies that are getting closer to human trials. The AACR 2026 meeting starts in about 20 days, and updates on ongoing clinical trials likely won't be available until just a few days before it begins.
Yeah, and I think we have an incredible lineup today. I mean, imagine a tumor building a literal concrete bunker around itself just to keep your immune system out. We're actually going to look at the exact chemical trigger that turns that concrete back into sand.
Which is mind-blowing.
It really is. But, before we get into that kind of microscopic preclinical engineering, we made a commitment to you, the listener, to always honor the clinical data first.
Right, absolutely.
We really need to ground ourselves in the reality of what is keeping patients alive right now, today.
I completely agree. I mean, we have to establish the baseline before we can look at the horizon.
Mm.
And when we talk about that clinical reality, specifically metastatic hormone-sensitive prostate cancer,
Right,
or mHSPC, there's a recent presentation that really puts things into perspective.
Dr. Christopher Sweeney's framework. Yeah. It's a vital perspective to have. If you look back at the 1990s, the standard of care for this advanced stage was androgen deprivation therapy, or ADT, entirely on its own.
Just the ADT.
Just the ADT. And historically, that yielded an 8-year overall survival rate of about 25%. So just 1 in 4 patients.
Wow, only 1 in 4. But fast forward to today, and that landscape has, it's fundamentally changed.
Oh, completely.
Because now, by combining that ADT with androgen receptor pathway inhibitors, the ARPIs, and
Sure.
sometimes adding the chemotherapy docetaxel, that 8-year survival rate has effectively doubled.
Mm.
We're all looking at 50% now.
Right. It's a monumental shift in oncology. It really proves that these combination regimens are not just, marginally delaying disease progression. They are legitimately rewriting the timeline of a patient's life.
But let me play devil's advocate here for a second,
Mhm.
because doubling survival is fantastic, obviously, but it begs a pretty procedural question.
Sure.
If triplet therapy, meaning we hit the cancer with ADT and ARPI and docetaxel chemotherapy all at once, if that is yielding these kinds of numbers, why isn't that just the default? Why not just hit every single patient with the absolute kitchen sink on day 1?
It's a logical assumption, right? But the biology of the patient and the toxicity of the drugs, well, they really complicate that strategy.
Ah, right, the side effects.
Exactly. Sweeney's analysis of landmark trials, specifically Charted and the phase 3 Enzamet trial, directly addresses this. The data clearly shows that ADT plus an ARPI should be the foundational backbone for eligible patients. But docetaxel is a systemic, highly toxic chemotherapy.
Right, it doesn't just attack the cancer. It takes a massive toll on the entire body.
Precisely. And the clinical data proves that adding chemotherapy for a patient with low-volume disease simply does not offer a survival benefit that justifies that kind of physical destruction.
So it's just not worth the damage.
Right. Docetaxel should be strictly reserved for patients with high-volume, highly aggressive disease. You really have to maximize the oncological strike while preserving the patient's physiological reserves.
That makes perfect sense. You don't use a sledgehammer when you need a scalpel. And speaking of measuring that oncological strike, those trials highlighted a very specific prognostic marker, didn't they?
Yes, they did. For patients on these ADT-based therapies, getting their PSA levels to drop below 0.2 nanograms per milliliter is absolutely critical.
Below 0.2.
Right. If your blood work shows that deep of a response, you are the one sitting in that 50% long-term survival cohort. Deep responses dictate long-term outcomes.
And alongside those chemical therapies, Sweeney also highlighted some survival signals coming from radioligand therapies, right? Like the beta-emitter lutetium-177.
He did. It's a modest but very real survival signal.
And that actually provides a perfect biological bridge to our next piece of clinical data. Because we know radiation damages cancer DNA, but, the human body isn't just a passive battlefield. We now have this fascinating data on how different types of radiation fundamentally alter the patient's immune system.
Yeah, this comes from a remarkable whole blood RNA transcriptomics study on peptide receptor radionuclide therapy, or PRRT.
Okay, wait, let's unpack RNA transcriptomics for a second for everyone. Because a standard liquid biopsy usually just looks for circulating tumor DNA, right? Or tumor cells.
Correct.
But transcriptomics is different. It's essentially reading the live diary of the cells. Like, it tells us not just what cells are present, but what instructions those cells are actively carrying out at that exact moment.
That is a perfect way to describe it. It maps the real-time activity of the immune system. So in this study, they looked at patients receiving either the standard beta emitter, lutetium-177, or a newer alpha emitter, lead-212.
And when they read the diary of the patients on the newer alpha emitter, what did they find?
The findings were incredibly structured. The alpha radiation didn't just kill cancer cells, it triggered a highly organized immune activation signature.
What kind of activation? Like, what is the immune system actually doing?
Well, the blood showed early, massive spikes in reactive oxygen species and really loud signaling for apoptosis.
Which is programmed cellular suicide, right?
Exactly. And on top of that, there was a heavy recruitment of monocytes. And monocytes are essentially the scouts and organizers of the immune system. They're the ones that call in a broader systemic attack.
So the alpha emitter is acting like both a bomb and a flare. It damages the tumor and flags down the immune system.
Beautifully put, yes.
So what did the transcriptomics show for the beta emitter then?
The beta emitter caused a profound state of lymphopenia. It aggressively wiped out lymphocytes, particularly B cells.
Oh, wow. So it just suppressed everything.
Yeah, pretty much. I mean, while both treatments naturally caused some collateral immune drop-off, the beta radiation was vastly more suppressive to the immune system's lymphoid compartment. The alpha radiation, on the other hand, successfully combined that localized DNA damage with a coordinated inflammatory immune response.
So the implication here is just massive.
Mm-hmm.
By checking a patient's blood using this transcriptomic profiling, we don't have to wait, 3 months for a bone scan to see if the tumor shrank?
Not anymore, no.
We can look at the blood weeks earlier and instantly know if the immune system is organizing a structured attack or if it's just getting wiped out.
Exactly. We are legitimately looking at the future of real-time predictive treatment monitoring.
Okay, so if targeted radiation and systemic drugs represent the external attack on the cancer, we need to talk about the patient's internal baseline. Like, how does a patient's daily lifestyle fortify their own biological armor against this disease?
This brings us to some very robust, highly scrutinized human observational data. Let's start with a massive international pooled analysis. They tracked over 12,000 men, specifically looking at the circulating levels of 25-hydroxyvitamin D in their blood.
And this was before diagnosis, right?
Yes, exactly, before they were ever diagnosed with prostate cancer.
12,000 men is a really heavy statistical sample. So what was the impact of the vitamin D?
The data revealed that men with higher pre-diagnostic vitamin D levels had a roughly 25% lower risk of prostate cancer-specific death.
25%.
Yeah. To use the exact clinical metric, there was a hazard ratio of 0.75 for an 80 percentile increase in vitamin D levels.
Okay, a 25% drop in mortality risk is huge,
Yeah.
But, and I have to push back hard on this, whenever I hear observational data about supplements or vitamins, my immediate thought is the healthy user bias.
Oh, of course. It's the classic confounder.
Right, because the guy who has optimal vitamin D levels is probably the same guy who, jogs every morning, doesn't smoke, eats a Mediterranean diet, goes to his doctor regularly. How do statisticians isolate the vitamin D molecule from the fact that this guy just lives a healthier life in general?
It is honestly the most critical question you can ask when reviewing observational studies. But the researchers were acutely aware of the healthy user bias.
So how did they handle it?
To counteract it, their statistical model strictly controlled for body mass index, physical activity levels, smoking history, and crucially, the clinical stage and grade of the cancer at the time of diagnosis.
So they essentially mathematically stripped those healthy lifestyle variables out of the equation.
They did, and even after isolating for all those confounding factors, that 25% reduction in mortality held firm.
That is genuinely compelling. It suggests the vitamin D itself is mechanically protective, not just a byproduct of a good lifestyle.
Precisely. And, we see a similar protective mechanical effect with diet. The health professionals' follow-up study tracked over 5,000 men for about 12 years, but this was after their prostate cancer diagnosis.
Okay, so post-diagnosis tracking, what were they looking for?
They wanted to see if diet impacted the risk of developing frailty.
And we should definitely define frailty here, because we aren't just talking about feeling a bit tired. In oncology, frailty means severe fatigue, sudden loss of grip strength, unintentional weight loss, things that can actually disqualify a patient from receiving life-saving treatments.
Exactly. It's a critical vulnerability. So the researchers evaluated these men using established dietary frameworks, specifically the DASH diet and the diabetes risk reduction diet.
And the results.
The men who strictly adhered to these dietary patterns had an 8 to 19% lower risk of developing clinical frailty post-diagnosis.
So we have clear human data that vitamin D, diet, and exercise protect the body, and extend survival. But, knowing that it works isn't enough. We want to know how it works. Like, how does a jog on a treadmill physically alter the microenvironment of a tumor?
And this is where we bridge perfectly from clinical observations into the cutting-edge preclinical lab data.
Right, because this is where the biology gets truly elegant. Let's talk about the tramp mouse model study.
Yeah. So researchers used a tramp mouse model, which is engineered to develop prostate tumors that mimic the progression of the human disease. And they put these mice on a consistent, moderate exercise regimen.
And going back to your opening thought, this is where that concrete bunker comes in, right?
Yes, exactly. So inside an aggressive prostate tumor, you have these support cells called fibroblasts. The cancer basically corrupts them, turning them into myofibroblasts.
Okay, myofibroblasts.
Right. And these corrupted cells produce a dense, fibrotic, scar-like tissue that physically shields the tumor from the immune system. It is literally a microscopic concrete bunker.
So how does the exercise break the concrete?
Well, exercise naturally increases the systemic levels of a specific cytokine called CNTF, or ciliary neurotrophic factor. The researchers discovered that CNTF acts directly on that tumor microenvironment.
What does it do to it?
It chemically forces those aggressive, scar-producing myofibroblasts to revert back into calm, resting fibroblasts.
So the exercise releases a chemical that turns the concrete back into sand. And once that physical barrier is softened, what happens?
The physical gates open. CD8-positive T cells, which are the apex predators of your immune system, they can finally infiltrate the deep tissue of the tumor and begin hunting the cancer cells.
That is incredible.
And they proved this mechanism, too. They took sedentary mice, injected them with synthetic CNTF, and watched the tumors shrink in the exact same way.
That is a brilliant mechanical explanation for why exercise works. But, here is the reality of oncology. Many patients with advanced disease are simply too frail to exercise.
Right. They just physically can't do it. So if a patient cannot generate their own CNTF, how are researchers chemically melting that bunker in what we call cold tumors, the tumors the immune system ignores?
Well, if the body cannot remodel the environment, we engineer a drug to do it. Enter MQI-201. This is a preclinical antibody designed to target a highly specific calcium channel on the surface of cancer cells called TRPV6.
Why target a calcium channel? Like, what does calcium have to do with the tumor's defense?
Prostate cancer cells heavily upregulate, or essentially hijack, these TRPV6 calcium channels to fuel their own rapid growth and survival. But healthy prostate tissue barely uses TRPV6 at all.
Ah, so it's a unique vulnerability.
Exactly. By blocking this channel with MQI-201, researchers didn't just starve the tumor, they saw tumor volumes drop by up to 90% in preclinical models.
90%, just from cutting off the calcium supply.
It's more than just starvation. Depriving the cancer of calcium creates massive cellular stress, and that stress completely flips the surrounding immune landscape.
How so? The number of tumor-supporting M2 macrophages in the environment dropped by 50%, while natural killer cells and T cells flooded into the area. It chemically forces a cold tumor to become hot.
Okay, so MQI-201 stresses the tumor into dropping its guard. But there's another preclinical approach we looked at that doesn't just stress the tumor, it acts like a Trojan horse. Let's talk about Bvax.
Oh, Bvax is fascinating because it leverages a totally different arm of the immune system. Instead of relying on T cells, researchers harvest a patient's own B cells. They activate these B cells in a lab, load them with specific tumor proteins, and then infuse them back into the patient.
And why B cells? I mean, if T cells are the apex predators, why send in B cells?
Because in immune-excluded prostate cancer models, the T cells get trapped at the outer edges of the tumor. They just can't get in. But the Bvax cells successfully infiltrate the deep tissue, bringing highly concentrated mHCI activity right into the core of the tumor.
Let's break down MHC2 for the listener. Because mHCT is essentially the immune system's biochemical wanted poster, right? It displays the chopped up pieces of the enemy so the rest of the immune system knows exactly what to kill.
That's a great way to put it, yeah.
So Bvax basically sneaks the wanted posters past the guards and hangs them up inside the headquarters.
That is exactly the mechanism. And by changing the architecture from the inside out, Bvax reduced tumor volume by 68% in those preclinical models.
So we have mechanisms to melt the bunker and Trojan horses to sneak past the walls. But what happens when remodeling the microenvironment just isn't enough? This is where we shift into the final hyper-engineered phase of our deep dive. I'm talking about the microscopic assassins, ADCs, DCs, and TCEs.
Yeah, let's start with the bispecifics. We have a new antibody-drug conjugate, or ADC, named ENOK-001. Now, a traditional ADC works by binding to a single target on a cancer cell and releasing a toxic payload. But ENOK-001 is bispecific.
Meaning it targets two things.
Right. It requires simultaneous binding to two entirely different targets, B7H3 and RRR1.
So it's basically a dual key system. It completely ignores healthy tissue that might only express one of those proteins and exclusively drops its payload on the highly aggressive chemo-resistant cancer cells that express both.
Exactly. But if a dual key system is effective, researchers asked, well, what happens if we use three? And that brings us to a trispecific T-cell engager called QLS-2401, designed for metastatic castration-resistant prostate cancer.
A trispecific, so it's binding to three distinct targets simultaneously.
Correct. It binds to PSMA and STEAP1, both of which are located on the surface of the tumor, and its third binding arm grabs CD3, which is located on the patient's killer T-cell.
It's like a microscopic biological handcuff. It shackles the T-cell directly to the tumor. But why grab the tumor at two different points, PSMA and STEAP1? Like, why isn't one enough?
Because cancer is fundamentally an evolutionary survival machine. If you only target PSMA, the tumor will simply mutate, stop expressing PSMA, and become invisible to the drug.
It just changes its disguise.
Right. By shackling the immune cell to both PSMA and STEAP1, you eliminate the cancer's ability to escape by just changing that disguise. It creates an inescapable grip.
That is brilliant engineering. But these guided missiles aren't just delivering standard cellular poisons anymore, are they? We are seeing entirely new types of payloads.
We really are. For instance, XYA02ADC is a new conjugate that targets a protein called MUC1C. And this is critical because MUC1C drives tumor growth entirely independently of the androgen receptor.
Oh, wow.
Yeah, this missile specifically hunts down castration-resistant and neuroendocrine variants, the ones that have learned to survive without testosterone.
And then there is the Degrader-Antibody conjugate called JZY2233. I need you to explain this because a degrader sounds fundamentally different from a standard toxin.
It is entirely different. Instead of dropping a poison that just burns the cell down, a degrader is a precise microscopic machine. JZY2233 targets PSMA to enter the cell, and then it releases a molecule that specifically hunts for the CBP300 proteins, which the cancer uses to drive its growth.
And how does it degrade them?
It attaches a biological trash tag called ubiquitin onto those growth proteins. This tricks the cancer cell into feeding its own vital engine parts into its cellular garbage disposal, which is known as the proteasome.
So it literally tricks the cancer into shredding its own survival mechanisms.
Yes. And because it's so targeted, a single dose in preclinical xenografts, which are human tumors grafted into mice, a single dose suppressed tumor growth for over 90 days without severe systemic toxicity.
Tricking the cell into eating its own engine is incredible. But what if the cancer mutates to evade the handcuffs and alters its proteins to avoid the degraders? There's one final preclinical mechanism we need to cover, and it involves exploiting the cancer's own emergency repair systems.
This is the concept of synthetic lethality. It's basically the ultimate biological paradox trap. We can see this in action with the MOS-101 clinical program, which is currently in a phase 1 trial testing a drug combination, ASTX-295 alongside olaparib.
Let's break down the mechanics of this trap, because this is fascinating.
So almost every human cell contains a tumor suppressor protein called P53. It is often called the guardian of the genome. If a cell suffers catastrophic DNA damage, P53 steps in and triggers apoptosis. It commands the damaged cell to die so it doesn't become cancerous.
But obviously in prostate cancer, the guardian isn't working.
Right, because many tumors upregulate a different protein called MDM2. Think of MDM2 as the jailer. It binds to the P53 guardian and destroys it. This allows the heavily damaged cancer cell to survive and multiply.
So how does the drug combination spring the trap?
First, you administer ASTX-295. This drug neutralizes MDM2. It essentially kills the jailer. Suddenly, inside the cancer cell, the P53 guardian is freed and begins to build up in massive quantities.
But just freeing the guardian doesn't kill the cell, right? You need to give the guardian a reason to pull the alarm.
Exactly. And that is where the second drug, olaparib, enters the picture. Olaparib is a PRRT inhibitor. It intentionally blocks the cancer's ability to repair routine DNA damage, particularly in tumors with BRCA2 mutations.
Oh, I see it. Olaparib steps in and causes catastrophic compounding DNA damage to the tumor. Normally, the tumor would just freeze its cell cycle, limp along, and try to survive the damage. But because ATX-295 has flooded the cell with the P53 guardian, The cell suddenly recognizes that the damage is fatal.
Precisely. The immense stress from olaparib, combined with the totally uninhibited p53 guardian, forces the cancer into immediate apoptosis.
Wow.
The drugs don't just kill the cancer, they force the cancer to kill itself. That is synthetic lethality, using two separate mechanisms to trap the cancer in a paradox it simply cannot survive.
So what does all of this mean for you, the listener? Let's take a step back and look at the massive spectrum of science we've just covered. We started in the clinical reality, looking at how combining therapies has raised the 8-year survival floor from 25 to 50%. We explored how the patient's baseline optimized vitamin D and targeted diets mechanically protects them. We saw how exercise releases CNTF to physically melt the concrete bunkers surrounding tumors. And finally, we zoomed down into the atomic level, exploring trispecific biological handcuffs, cellular trash disposal hijackers, and the synthetic lethality of trapping a cancer's own DNA repair system.
It is a breathtaking acceleration of targeted science. It really is. But, as we engineer these perfect biological traps, it leaves us with a fascinating evolutionary question.
What's that?
When we force a tumor into a corner with trispecific antibodies, degraders, and synthetic lethality, are we actually curing the disease? Or are we just hyper-accelerating its evolution, forcing it to become a highly manageable chronic condition that a patient lives with for decades, much like HIV is managed today? It's a profound psychological shift that the oncology field and patients themselves will have to navigate in the coming years.
Stay strong and fight on.